I grew up in a large family. I have 7 brothers and sisters, and we lived on the outskirts of Fort Worth in a big area. We had about an acre, so we had places to roam. Next door neighbors had horses, so of course we were outside all the time. There was no TV or certainly no way to keep ourselves occupied otherwise. And um, also, my grandparents had a farm which was about 20 miles away, and we went out there on the weekends, and I spent my summers out there. And this was an old-fashioned farm that had lots of woods, a few fields, and lots of cows. And so I learned to just be outside so much. I loved it. I loved being outside, outdoors. And I think, that made me think maybe I would like to be a scientist or something. So that was my background. Well, when I was in high school, I wanted to be a scientist. I envisioned myself in a white lab coat and chemicals bubbling up and down. And when I got to my undergraduate, I found out that I really liked biology better than chemistry. I loved the study of biology. And so I continued to do that for my undergraduate degree, and then went to UT Arlington for my master's. And the program there, I was working with a mammologist, and I did for my research a study of homing behavior in cotton rats. And we'd catch these cotton rats and mark them and take them off half a mile, different areas, and see how fast they came back to get caught in the traps again. And it was an interesting finding, that the young juveniles didn't return very much, but adults, and particularly mothers with young, came back from up to a mile immediately to get back home. So that was interesting. But I thought, gosh, I'm not going to get a job with studying cotton rats, or at least any job that I wanted. And I was offered a position to come to Texas A&M University and work with the fisheries people there. And I decided that was a much more reasonable thing to do. And so I did do that and got very much interested in studying the marine fishes. We worked in Galveston Bay, and particularly early life stages. So I worked with juvenile small fishes and larvae, and that got me interested in fish and larval fish particularly. Well, it was certainly an interesting experience when I went to Texas A&M in fisheries. I was the only female graduate student in fisheries. There were a couple in wildlife, but it was mostly males. And so I learned very quickly to get along with all these guys. We worked together and did a lot of studying together, and it was felt very comfortable. And in fact, I met my husband there, who was working with us on the projects that we were working on at A&M. So that was a very positive result of being in an all-male field. When I went to UTMSI, again, there weren't a lot of women. I worked with Dr. Connie Arnold right away, working with the redfish issue, red drum, and it was very, very comfortable. But as I moved up, it became, I think, interesting to realize that all the faculty were men, and all the, I guess, teaching was done by men, except for me. I've always had gotten along with them, but it, I think, makes you stand out, and maybe women have different ways of approaching subjects and different ways of moving forward that are sometimes appreciated and sometimes not appreciated by the dominant men in the society. I think now it's becoming a little better, but even in science, there's still very few women compared to men at the upper levels of faculty. I would like to see that changed, and I'd like to see more women involved, and then I think it would change the perception of of women scientists or women professors a great deal. When I first started to work at UTMSI, I was working with Connie Arnold, and he had just figured out how to spawn redfish. And they had all these eggs and larvae, and my job then was to figure out how to raise these things. My very first job took me a year to figure it out. I mean, the larvae are so tiny and so small, and they can't see well, and they can't swim well, and they need plankton. So that's what they eat in nature, is zooplankton that's in the water column. And so I mixed algae and rotifers, which we could hatch out and grow in the lab, and tried to get them to feed down on them. I had to worry about the temperature, the water quality, that was particularly important, and salinity, of course. And I can remember trial after trial after trial of trying to keep these things alive. And it took me about a year to figure out exactly what it was that I need, how good the water needed to be, and the salinity, temperature, and feeding regimes for these guys. I was very rewarded by being able to keep them alive until they became juveniles. And that took about 3 weeks or so for them to grow well enough to get their fins, to get their scales, to get their development of their stomach and the digestive system, so that then they could act like a normal fish. And really, this started me on my way with my interest forever after then in the early life stages of fish, knowing how sensitive they were and how difficult they were to raise. Nowadays, everybody can raise redfish. It's pretty easy and pretty fast, but then it took a long time to figure it out. Well, now, I guess people know what it is that they need to feed upon. They know when they need to feed, and they know how often. They know that temperature is important, salinity is important, and put those parameters very much in line with what their spawning parameters are, and they could be very successful. And this was finally transferred to the state fish hatcheries, and they then put those larvae out into ponds that they could produce lots of zooplankton in, and the larvae could grow up in those ponds and big enough till they could release them into nature. At the Fisheries and Mariculture Lab, which is about a mile across from the main UT Marine Science Institute, and it was an old, I guess, fish, what was it? A fishery, National Marine Fisheries Lab is what it was. And then they closed that, and it was turned over to UTMSI. And so Connie Arnold was there working, and he had just figured out how to spawn redfish using temperature, photoperiod, and the seasonal cycle that normally occurs in nature. He just said, okay, they're supposed to spawn in late summer, we'll put them through this whole cycle, and when it comes to the right time, they'll spawn. And of course they did, which was, nobody had thought you could put a great old big fish like that in a tank and get it to spawn naturally. But he did. And one day we were working, and we heard this helicopter, and it landed right out in the field. And it was Perry Bass. And he came in saying, hey, I hear there's somebody here raising redfish. Who is that? And so we met Perry Bass, who loved redfish, and he was working very hard with the group, Gulf Coast, what was it called? CCA now, but it was Gulf Coast Conservation Association was what it was called. And he was working with them to make redfish a sports fish only. At that time, commercial fishermen were using nets and collecting all these redfish for restaurants and for sale to grocery stores. And they wanted to change that because the populations were so low and going down, and there was such an important sports fish. And so he came and was asking what we were doing and saying that we could use, if we could do this, maybe somebody could grow them in captivity and sell them to the restaurants and to the grocery stores, which is eventually what happened. Someone several people were able to do that. And so that was an interesting and exciting experience. And Perry Bass became a great supporter of ours and set up scholarships and fellowships, and provided for a lot of research on red thumb. I knew that there was a big ornamental aquaculture, ornamental commercial facilities going on all over the world. People were going out and collecting fish from the wild and selling them to the aquarium trade. And it was becoming more and more popular that people wanted to have marine aquariums with these beautiful fishes. And the only way to get them at that point was to go collect them. And people began to worry about over collection, changing populations, picking certain species or certain, I guess, looks that they wanted, and that was going to affect what was left of the population, or maybe even deplete the population. And I had been out snorkeling and well, along with other people around the jetties at Port Aransas, and seen some tropical fishes there. And we thought, well, maybe they would react the same as redfish, spotted sea trout, croaker, all the species that we had been able to raise. Maybe we could bring in these beautiful tropical fishes and produce them so that we could figure out how to do it and let people begin to grow them instead of collecting them from the wild. So we started out by collecting fish there at the jetties and in the seagrasses. We had cubby ewe, we collected seahorses, wrasses, several species that we could just find there, or go out on the oil rigs, and around those oil rigs there would be these tropical fishes along with the snapper and other species that like to congregate there because there was food and protection in those oil platforms. And so that really began the journey that I started trying to use the same I guess procedures that we had for redfish. Control the temperature and the day length, manipulate that until it looked like what the normal spawning conditions were. And for these tropicals, it was of course warm and long day lengths generally. And then after the eggs were produced, they were pelagic like redfish eggs, like most marine fish. The eggs just float to the surface and there's no maternal care whatsoever. So we would collect these eggs in special collectors we made in these broodstock tanks, and then try to raise them. And that was a real challenge in many, many cases. Some would feed on the rotifers, just like the redfish and trout had done, and others would not. And we would have to collect wild plankton to try to feed them. In some cases, we tried to culture copepods, and we could do a few of those and feed the larvae and get them through. We could do that with the seahorses. That worked pretty well. And some of the little fish we never could raise at all. And of course, I suppose that's because we couldn't find the proper food for them. But it was a fun experience. A lot of my students got to work on these marine tropicals. And we tried to just work on those that are found within the Gulf of Mexico and the Caribbean. So we didn't want to go very far away, but pick those species that would be important for these areas. There were a lot of organizations that then were having meetings and get-togethers for marine ornamentals, and were always wanting to know what we had found out and what the new findings were, so that these fish could be raised in captivity. And now there are several species that are being successfully raised and commercially sold. And so that makes me feel really good. But in the meantime, we also learned a lot about the larval stages that weren't known of these different species. So that was interesting scientific information as well as practical. I think that it really has helped change the mind of people, thinking instead of having some real exotic, hard to find deep sea fish, Now they say, okay, these are being bred in captivity. They're beautiful, and that's what we want. It's sort of like the green revolution, in fisheries. I eventually retired in 2010, and then I was asked to come back for 1 year to act as acting chair of the department as we were going through a transition. And I did that for a year, and then I was asked to come back to work as acting director because we had just lost our director. And I agreed to do that, but I said only half time. And so I went back, and we eventually hired the director, and I was able to finally retire. But it was it was fun. I enjoyed it a lot of hard work, but the students and the people were the rewards. I remember it was exciting going to work every day because it was always a new challenge, and you never ever solved completely everything. When you learn something, that just opened the door for something new. And so I never dreaded going to work. That was a lot of fun, and getting your hands wet and keeping the seawater on the floor. Yeah, that was a lot of fun to do that. Some of the work we did is still important. I did some work for the Water Development Board to look at the effects of changing salinities on the early life stages of croaker, trout, and red drum. And that was because of the changes in freshwater inflow into the bays, how that was going to affect the populations of fishes. And even today, that's being used for looking at desal, that's using seawater, and the discharge of high salinity water, how that might affect the early life stages of these important species that are still very, very important to the sports fishing trade. I think I'm most proud of all the students that worked with me and that I was able to influence and help and support so that they became active scientists or teachers and took those lessons with them as they went forward. I loved working with graduate students. It was a real pleasure for me, and to see their success was just wonderful. One thing is interesting is that I was the first one in my family to go to college and get a degree. And I was really pleased that 1, 2, 3 of my sisters followed in my footsteps. One became a teacher, one became a lab tech and phlebotomist, and now she's in charge of a whole blood center. And another one became an ornithologist, and she became an excellent leader in various places in Texas, the Gulf Coast Bird Observatory, and has now retired, but she influenced me too for birding, so I've become a birder as well. So I was really pleased that my sisters were able to succeed as well as myself.